Okay, in November of 2023, NHGRI sponsored a workshop titled Advances in the Architecture of Complex Human Traits. Alexander Arguello is the lead PD from the Division of Genomic Sciences. And he was the leader in organizing that workshop. I think that's a fair statement. And Councilmember Nancy Cox was an attendee there, so they're going to tag team a report on that workshop.
So thank you, Rudy. So as Rudy mentioned, this is a workshop that we had in the fall. It was a 2-day in-person workshop here in Bethesda. And we're just going to give you a very brief and a high-level overview of some of the highlights of the meeting. But I do want to point you to the workshop on genome.gov, the website for the workshop. Here you'll find the full meeting agenda, full list of participants, a written meeting summary by us, as well as recordings of all the lectures, discussions, roundtables you'll find on the website. So if you're interested in more details about the workshop, please visit this resource. So the workshop was really about complex human traits and understanding their genetic architecture. And by genetic architecture, we mean the number of alleles that are contributing or affecting a trait, what is the frequency of those alleles in the populations, how those alleles interact, and what is the general effect sizes of those alleles. Now, now that we have sort of large-scale genetic data in these very large cohorts, we're able to sort of quantify some of the parameters that describe genetic architecture, from oligogenic traits, which involve a handful of moderate to high impact alleles, to highly polygenic traits that involve many small effect alleles. And we are able to sort of extract general principles about the types of traits that are highly polygenic, such as brain and behavior-related traits. But it's important to realize that genetic architecture is like heritability. It really depends on when and where and what population you're characterizing the trait. So there is this larger context and non-genetic factors that influence genetic architecture. And it has really implications for how we design our genetic association studies, for gene discovery, how we biologically interpret those results, and what is sort of the tractability of different traits in terms of different types of intervention. So it's sort of within this context of why we looked at our strategic vision, which really highlighted the need for considering the genetic and non-genomic contributors to health and disease traits, as well as one of our compelling genomics research projects. The slides look a little wonky here. But really to help us, one of the goals was to determine the genetic architecture of most human diseases and traits. And this sort of motivated us to release a funding opportunity recently that this council is aware of, really seeking novel approaches, methods, statistical, computational, mathematical approaches for understanding genetic architecture, especially integrating non-genetic and genetic factors, as well as looking across levels. And this is a standing announcement out now with standard NIH receipt dates, and council may be hearing about some of these applications at the next council in the fall. But this really sets us up nicely to address some of the questions that remained after the workshop. So the workshop was organized by a scientific organizing committee, including Nancy, as well as Lauren Crawford, Molly Pishvorsky, and Peter Vischer, that were instrumental in putting together a scientific program and really identifying a diverse group of investigators from across career stages that made key intellectual contributions to the workshop. And this was a very highly motivated, engaged, talented group of people that made the workshop quite stimulating. So a little bit more about the workshop participants. We had about 66 participants. Most of them were here in person in Bethesda. Some of them joined us remotely online. And although the focus of the workshop was on human traits, we did want to include people that worked on a diversity of species. And actually this perspective was actually very valuable in sort of contextualizing some of the issues and challenges that we faced in understanding the human traits. And we also aimed for geographic and regional diversity in our participants, including people not only across the US, but other places of the world where they're making intellectual contributions to genetic architecture. And I should mention that most of the people, of course, worked in genetics, from population, quantitative, evolutionary, statistical genetics, but we also wanted to include people in adjacent fields, especially in the social sciences, people in anthropology, psychology, economics, sociology, and epidemiology, given the broad scope and sort of perspective that we need in understanding fully genetic architecture. And we had a lot of interest from researchers and the public who joined us online, over 500 attendees. So the objectives of the workshop were to really adopt a historically grounded and forward-looking approach to genetic architecture. We have over a century of quantitative genetics, and how can we contextualize sort of the large-scale genetic and genomic data we have given that. We wanted to sort of assess and build upon the current state of the field, as well as identify gaps and potential emerging opportunities, right? What data are we not collecting? What are these questions that we aren't asking? And also explore sort of future research directions, especially given all this new data, integrating across biological levels. And finally, we wanted to see if we could facilitate a theoretically informed universal approach for understanding complex trait architecture, right? Given all the different types and varieties of traits we're looking at, is there one sort of framework that we can approach it to understanding all these different traits? So the meeting was sort of revolved around scales of biological organization. So we had a series of talks, keynotes, discussions, and roundtables, all sort of revolved around scales of biological organization. So at one end, we had cells, tissues, and organs, and this was really about linking genetic variation to lower level molecular cellular features, using data from large-scale functional genomic assays, and reconciling the variants that you identify for those with sort of the variants that you see with these higher level organismal traits. And sort of one of the take-homes from this session was that even for these simpler systems, the genetic architecture is still quite complex. And then we had anatomy, physiology, and behavior. And these are sort of the types of traits in which quantitative genetics really cut its teeth on. For example, height. And so height emerged as the model complex trait. And some would argue that the genetic architecture of height is almost nearly completely resolved. But to what extent can we extract principles from height and apply that to other more complex traits? There's also some discussion about traits that may have sort of the same heritability, such as some disorders of the gut, some disorders of the brain, and how those can nonetheless still have diverging genetic architectures. And finally, in families, populations, and societies, really looked at these traits and how they may be affected or influenced by the people around you, especially people that share alleles with you, as well as the meaning we attach to or the way that we measure traits in different social cultural contexts, or the way that these traits depend upon how we organize societies. And this is important for how we biologically interpret findings about the genetics of traits. So again, this is just very high-level overview of the workshop, but again, I point you to the meeting website for further details. Now, Nancy will go over some of the highlights of the major scientific themes that emerged from the workshop, some of the outstanding questions that we had after the workshop, and so sort of the wider impact that was felt after the workshop. So, Nancy.
Yeah, so a key emerging theme was the population and statistical versus the biological and mechanistic. Some of the age-old challenges and different perspectives that people bring to these kinds of studies. Clearly, it's obvious. This kind of framework difference would change what you see as the needed next steps. And there were really interesting, it was great that we had the model systems, because I think some of the observations brought in from the model systems really helped us understand better some of the ways that we might have been misled by model systems findings. The largest effect sizes from some of the model systems really did come from natural selection for traits in, if you think of animal breeding, virtually all of the large effect phenotypes involved alleles that, had arisen naturally, but were bred in very deliberately and would never have gotten to the observed frequencies otherwise. They were dragging some things that would have been selected against had it not been for the very deliberate breeding. So some of the differences between what we see in these, in certain kinds of model systems versus what we see in humans reflect those kinds of differences in the actual breeding models. And so we see, confounds between the population level and individual level inferences that we might draw in those contexts. The G by G and G by E that we sometimes see much more readily in model systems are also, again, part of the consequences of inbred lines versus outbred populations. And it's not that we didn't know these things before, but the relative contributions of the breeding systems and selection haven't always been so apparent as what we really looked at the meeting. Really, context matters. It came up over and over again in the meeting as one of the things that is a differentiator in how we think of effect size. When we estimate effect sizes in ignoring context, and so what we're looking at is something averaged over all contexts, and whereas in the right, often unmeasured context, effect sizes may be much larger. For when we think of polygenicity, you saw that graph at the very beginning. Context for BMI may be quite different than context for height. Protein may be a big driver of height. Polygenicity, we know that it can be an important factor in people achieving their highest possible genetically determined height. In animals achieving their largest possible growth parameters. But BMI has height involved, but clearly different kinds of factors that would affect the ultimate BMI achievement. And context really varies by the level of biological organization. And when we talk about context at the level of multiomics, the transcriptomics, proteomics, et cetera, there's so many contexts that may matter. And whether we talk about context specificity, maybe it's not specific to particular things. There may be many things that can affect context. But the context, the ways that context can affect what we measure is clearly important. So cell type, population, and the ability of that to vary by time and place, by the age of the organism, that level of context is clearly important as well. There are probably genes that matter much more in early development than in later life, and in some cases we can't really go backwards for that. Exposures that modify genetic effect size, that modify regulatory networks, and there was some really nice, sophisticated discussions that looked at the ways that regulatory networks may become disorganized, that the main feature may be not at the level of the gene, but the disorganization of the network itself, was also an important emerging discussion point, I think, that came up at the meeting. And so, yeah, integrating across contexts, perturbing context to try to capture more of the information on context is clearly a major focus of the field. Also, very different complex traits have just different levels of complexity, different opportunities for complexity. And so dividing traits along biological versus social axes may not be so productive. The ways that different traits can be confounded in association studies was also brought out very nicely by some examples that were provided. And I think will continue to be something that people focus in on for some of the traits that have relationships to socioeconomic status, for example. I think the economists brought in some interesting perspectives. And so another key point was the need for methods and study designs that can adequately account for different types of biases and confounding. And I think that will be a key point going forward in terms of the different approaches to methods. Open questions. It was brought up more than one time. It's like the current population genetics models being used were developed more than 100 years ago. And the question of whether everything that we need to go forward is really the right stuff came up more than once. And I think some of the work Molly Przworski has brought forward with respect to what we thought we knew about mutation and what all of the new data actually tell us about mutation and how it occurs and what's important. It is a really key set of observations reminding us that we need to revisit what we think we know with all of the data that we actually have now. And so some aspects of population genetics theory will ultimately need to be revisited as we really go through all the data that are being collected. All the sequence data that's available now really informs us on a lot of aspects of theory in a way that we just never had the data for before. And so some disagreements that existing approaches may have reached their limit, especially for the most complex context-dependent traits. How do we precisely quantify and measure non-genetic factors? So the importance, heritability as a concept is time and place dependent. The importance of really measuring non-genetic factors, and even when we can't directly measure them, are there ways to identify their consequences with measurements like epigenome, patterns, epigenome measurements at scale. The genome is discrete and finite, the environment is not. There are going to be many more ways that the environment impacts traits than we appreciate, and as environments change, heritabilities will change. there was a lot of discussion about what level of granularity and specificity is actually needed. The ways that multi-omics could be brought to bear on these questions, I think, was a big area of discussion, and certainly the perceived value of having much larger samples with epigenome measurements of a variety of sorts, I think was universally agreed would be a good thing. And how the non-genetic factors may or may not reveal unique biology. I mean, you could imagine that maybe environments, the biology of what environments drive layers onto what we're already learning from the genetics, or in some cases maybe largely orthogonal, reveal new biology that could be the target of completely different kinds of therapies and interventions and preventive measures. And so the need to learn more about what aspects of environment actually drive biology, so really create their own long-term biological consequence, like epigenetic changes that change gene expression over a full lifetime. What kinds of environmental exposures are so ubiquitous, they may not need to drive biology at all while still influencing long-term health outcomes. So I think the ways that exposures are can be over an entire lifetime without driving biology versus those that are clearly driving long-term biology, I think was another interesting discussion. And I would also note there was a reasonable amount of history to this. So there was a meeting actually sponsored by NIGMS some years before on genetic architectures, and there were a number of people at the meeting who had been at that much earlier meeting. And it was a little distressing to realize that how much we missed about what the challenges in genetic architecture would be. That is to say, what we thought we needed in the, whatever it was, late 1990s at this meeting, we achieved all of those goals. We achieved all of that discovery. But we would never have imagined that we would get to this level of discovery, thousands, many thousands, tens of thousands of DNA variants highly reproducibly associated with complex traits, complex disease, and know no more biology than we actually do from that. Because we couldn't conceive of the possibility that you would have these discoveries without knowing what the genes and other functional units were. That was inconceivable to us and it's not that we didn't know that there would be regulatory variation. We just didn't imagine that we would not have genes as anchor points with a lot more coding variation to any of these traits. And so there was a bit of a side exercise in discussions throughout the meeting about where our blind spots would be today. And I think, so Jennifer's talk really touched on a couple of them that are relevant for rare diseases as well. And this whole RNA world was one of the possibilities for blind spots that we haven't appreciated at the level of both additional RNA functional units, but also things like splicing that splice isoform ratios probably matter, and we're only now, with long-read RNA-seq, able to even look at that at scale. And we've always known that could be a big deal, and especially in tissues like brain, where we know there's a lot of splicing, really getting a handle on how more common variation may impact splice isoform ratios could turn out to be a big deal in helping us understand more about complex trait and common variation. But I think it's a good exercise and remind ourselves that we can can't really think past our ignorance on things that we don't fully understand. And so what pieces of genetic architecture we won't appreciate for another 5 or 10 years is it's a reminder of how far we have to go to really understand all of this. So I would say also it was a very there was a lot of side discussions at all of the breaks, even the meals, which people were sharing at the hotel. I think a number of collaborations have already come out of the meeting, things that people would like to try to look at sort of across these spaces of theory versus data generation and different populations. I think there are several meetings following Cold Spring Harbor and a Columbia Precision Medicine meeting that now includes a number of different groups that were present at this meeting, carrying on some of the conversations that were started here. So it was from many perspectives, a very provocative meeting, and a lot of fun to get to a second meeting on genetic architectures, where, I can hope that what we think of now will actually be the right things, but good to remember. It's hard to see past your ignorance to the things that we don't even know about yet. Yeah. So thanks to all the participants. There was a really good group of participants, very active, lots of discussion, and our colleagues, I think Molly might be on, and Lauren, and please jump in with any other observations that you would have.
Questions? Judy.
Yeah, Nancy, profound as always. So speaking of blind spots, the two blind spots I think about all the time is the sparsity of single-cell multiomic data. And that's kind of one thing. But then the other blind spot is our incapacity to scale protein analysis. And so can we think of kind of systematic approaches to scale and impute across these blind spots that we have?
So certainly the paucity of cell types that have been able to be investigated at a detailed level was a big part of the discussions, and a major part of context that everybody appreciates is really critical. And I think, not for sure solutions to any of it, but some discussions of the ways that we might use cell-based ontologies to be better organized, about trying to hone in on at least all of the major developmental ontologies that really need to be fully interrogated. And I think, all the different kinds of macrophages, for example, really come to mind. Microglia are critical in Alzheimer's, but osteoclasts and all the macrophages in the gut, it's a really key piece of biology that is relevant in immune biology, relevant in wound healing. And so, so how many kinds of macrophages do we need to interrogate? Or, really, do we just need to interrogate a small proportion of the types of macro? And context in activated versus inactivated versus underactivated and overactivated. We need those contexts as well to, I think, fully appreciate all the ways that macrophages participate in disease and ways that genetics will affect that regulatory biology. So the cell type, couldn't agree more. It was a major point of discussion. Your other point on what?
RNA
Oh,
to protein.
yeah, that's, I mean, that's a hugely important technical issue that that's technology development that is absolutely required. I think there's a lot of optimism, even in the technologies that we have now. They're really driving some good discoveries when you can see them at scale, as is available in the UK Biobank, and will be more and more available, I think, as some of the studies NHGRI is funding, where they're really able to do multi-omics, yes, on small sample sizes, but I think we'll learn more about how it all works together. But we're probably just, I mean, it's going to be a while before we see proteins in the way we can see other things. Did Molly or Lauren have any further comments? I don't know if they could even tell us that. Because they're probably just looking at.
Okay, Xander, Nancy, thank you very much. We'll move along.